Now I'd like to move on. We're moving on to Dr. Fran Priddy. I'm very excited to have her join us. Dr. Priddy is from the Malahan Institute of Medical Research. She's a clinical director there of the Vaccine Alliance Aotearoa, New Zealand, VANS. Dr. Priddy received her medical degree from the University of California, San Francisco, and holds a master's in public health from the University of California, Berkeley. She completed her medical training at Brigham and Women's Hospital, Harvard University in Boston. In 2006, she joined the International AIDS Vaccine Initiative, where as Chief Medical Officer, she led clinical strategy and epidemiology for over 20 viral and bacterial vaccine and antibody candidates, including HIV. And now she's the clinical director of Vaccine Alliance Aotearoa. So I'm very glad that she's able to spend some time with us this afternoon and share what's happening in that space in New Zealand. So welcome, Dr. Priddy. Thank you.
Thanks for that nice introduction, and tēnā koutou katoa. Just learning my Maori since I'm new to New Zealand in the past year. So thanks a lot for the invitation, and I've enjoyed learning more about HIV in New Zealand today. Actually, this is probably the most I've learned about it since I've been here, since it's been so focused on COVID. So what I wanted to do today was give an update on HIV vaccine research. Is there, are you going to project the slides, Mark? Okay, make sure I can see them a little bit better. There we go. Thank you. Okay, and are you can I control them? Okay, thanks very much. Okay, next one, please. So I just give a brief background about me, because you might help to know where I come from, since I'm obviously not a Kiwi. And my background is in adult infectious disease. And I trained in the US, but I really had an interest very early on in international health, and started out actually in maternal child health. And that's a picture up in the corner of some beautiful ladies from the Congo, used to be Zaire. And I was very interested in women's health, maternal child health in Africa. But very quickly that became, the HIV epidemic got into that population, and my interest transferred over to HIV. I did training in the Navajo Nation in the US. I have a lot of interest in sort of disadvantaged populations or marginalized populations. Again, another set of beautiful women from that culture. I did my training in Atlanta in infectious disease, and as Mark said, have then spent most of my time really in the past 10 plus years doing vaccine development for HIV vaccines specifically. So trying to find an HIV vaccine, which is what the organization IAVI is focused on. And can you go to the next slide, please? I came to New Zealand a year ago, though, to help this group, this VANS group, which is funded by NBIE, but it's based at the institutions shown there. I'm at the Malaghan, but it's a collaboration to try and do vaccine development for COVID vaccines in New Zealand, so that New Zealand can have those capabilities. So it's great to talk about HIV vaccines, though, with this group, because that's actually my main passion in my career. Okay, so next slide, please. So I'm going to talk about, do we still need a vaccine? Why is it so challenging? What's in the pipeline? I'll talk a little bit at the end about passive immunization, meaning injectable antibodies as a kind of bridge until we can get a vaccine. And then I have one bit on COVID vaccination for people living with HIV, if just an overview, if that's helpful. Okay, so do we need a vaccine? Next slide, please. I think this is a slide from 2018. And even then, this phrase, the end of AIDS, cure, ending AIDS, et cetera, was all the rage at the IAS meeting that year, and it's continued on. And it's an important phrase and goal to end AIDS. And actually, a cure is also important. And now that we know more about virology and have a number of treatments, the potential to cure HIV is doable in the future. However, we're not really going to be able to end AIDS without a vaccine. As you can see from COVID, which is a very different virus, we really can't even control it we're having a hard time with the vaccine controlling it. So the end of AIDS is going to need vaccination as part of its plan. And we also need vaccination until we get to a way to end AIDS. So the next slide, please, is showing what it would look like. Oh, sorry, I forgot about this one. I just wanted to mention these are some of the other, obviously, prevention methods besides a vaccine. So there you have our sad HIV vaccine at the bottom, chugging along over the decades. It's been 30 plus years now, no vaccine. But, you know, people are creative, smart, science moves along, and we've developed a lot of other ways to prevent HIV. I'm just showing some of them there, and when they've been approved is the red stars. So the piperine ring has a regulatory approval, long-acting injectables, cabotegravir that had positive data recently as a long-acting antiretroviral for treatment, but also for prevention, should be coming up for approval. And there's another long-acting one that's actually even more long-acting, 6 months, that's in the pipeline. So a variety of different types of PrEP. So the next slide is trying to show you, despite all this great progress, we still do need a vaccine. So on the left is showing how many new HIV cases would we have, new infections would we have by 2070. So we're looking long-term with no vaccine, but we did all the things we can do now to prevent, if we really ramp them up and put the money and the impetus there and really ran those programs as they should be run and really got out there, and many people were using PrEP, et cetera, we would have by 2070, a vaccine would help us, but with no vaccine, we'd still have a number of quite a bit of infections, 16 million infections. But if you look to the right, that's a more realistic picture. This is what probably will happen. The current trends of PrEP use and uptake of other prevention methods may improve a little bit, but you know, we know it may not get much better. So then there's going to be 40 plus million new infections. If we had an effective vaccine, we could drop that down to what it looks like on the left. So it's just showing there is a huge space where we still need a vaccine despite all the other methods that are coming along. Okay, so why is a vaccine so difficult? Next slide, please. Well, I just wanted to mention COVID, and I'll talk a little bit about how HIV is different than COVID. You know, COVID's a devastating pandemic. COVID's a zoonotic disease coming from the animal world. You know, HIV was in our lifetime the original devastating pandemic, also a zoonotic disease. And this is showing AIDS deaths globally on the top over 40 years, and this is showing COVID deaths on the bottom. So if we had this pandemic continue every 2 years at the same rate, we would end up with more deaths than the HIV epidemic over 40 years. But you know, we think the pandemic, we're going to be able to control it. So probably the impact of HIV, you know, and the number of people dying is higher overall than the current pandemic. I mean, that's kind of obvious from this, but just to try to put it in perspective, they're both critically important, devastating in different ways, different timeframes. Okay, so next slide, please. So why has COVID vaccine development been so successful? And I'm going to compare that to HIV vaccine development, which has been spectacularly unsuccessful. Not to say there's not a lot of good science, but it has not been successful. So the main reason is the virus. The COVID virus, SARS-CoV-2 is a relatively stable genome, and I'll show an image to describe that. Even though we have variants, et cetera, it does not mutate like HIV does. We develop natural immunity to COVID. We develop antibody responses, T-cell responses, and people who survive clear the infection. We have a neutralizing antibody response, and we know that that spike protein is a good target for vaccines. And then the epidemiology is different. It's a respiratory spread virus. It can affect all ages. So it does not have specific populations that can be stigmatized or ostracized. I mean, yes, there has been stigmatization within the COVID epidemic, but nowhere near as what was seen with HIV when you had, because it was sexually transmitted or through intravenous drug use, for example, you would have specific populations that could be stigmatized. So there was not the global emergency, that sense. The scientific moment is very different for COVID. We've had several decades now of vaccine development with next generation technologies. And then COVID came in to the picture at the time when we had some pretty amazing technologies, genetic sequencing, viral sequencing, many other technologies that could be quickly applied to it. HIV came along at a much different scientific moment. And then the political and financial investment and will has been pretty obvious that because it's a global epidemic and because it impacted across the board, countries were able to put, and it was such an economic impact. You had the incredible amount of resources, government cooperation, et cetera, that can make COVID and with pharma also a very key player to make COVID vaccines successful. And HIV has had very, very up and down levels of political and financial commitment over the years. Next slide, please. So this is a graphic to try and show you where does COVID fit in terms of viruses, in terms of its genetic diversity. How much does it mutate? So way on the left, you can see SARS-CoV-2, and the size of the ball is how diverse is the virus, each virus family, basically. So SARS-CoV-2 is not diverse at all, even though we're seeing variant development, et cetera, compared to on the right, the big green balls. HIV, influenza, and HCV are very diverse human viruses that infect humans. And so the numbers 1 through 5 on the top are showing which viruses we have effective vaccines for. And you can see the ones where we have effective vaccines like Ebola, which is shown in the blue, mumps, hepatitis B, measles, those are all highly effective vaccines. Those generally are really not very variable. And that's one of the main reasons why it's been possible to make a vaccine for those infections. And it's been so difficult for the ones on the right. Influenza, yes, we have a vaccine, but because influenza is so variable, it's not a very good vaccine. We have to update it every year. Next slide, please. So HIV, if you contrast that, the main reason it's been unsuccessful, the vaccine development, is because of the virus, because of the genetic variability. Natural immunity is not known to occur. We do know there's a spike protein on the HIV envelope, and we're very familiar with it. We understand it. Being able to neutralize that has shown that you can prevent HIV if you can neutralize the envelope, but the body's response to the envelope is polyclonal. It has many different responses because the envelope's constantly mutating its coding. And so that's a huge challenge. And then the epidemiology, like I mentioned, it is primarily as an STI originally, really led to stigmatization and didn't get that global push to make a vaccine. And then the scientific moment I mentioned was very different when HIV came around. And vaccines at that time were really more focused on childhood diseases, generally speaking. So this idea about developing a vaccine for adults, although we had the flu vaccine for elderly, et cetera, but this idea about developing a vaccine for young sexually active adults was just not, this was prior to HPV vaccine, many other vaccines. So different time. And then we just did not have the political will. It's come and go. We've had some great times of political will and funding and some not great times. Next slide, please. So I don't know if you can click, Ron, if you're doing this, if you can click on the bottom. This is a cartoon that will play if you click on the black space. Okay, that's fine. So basically, this is the spike protein for HIV. It's the HIV envelope glycoprotein. And all of those purple little balls, those are basically the sugary glycoproteins that coat the envelope. And there's multiple envelope spikes on every HIV virion. And that's the target for neutralizing antibodies. But the problem is those purple spikes, every time the virus copies itself, it makes mistakes. And a lot of the mistakes are on those purple glycoproteins. So we might get a vaccine can get a great neutralizing antibody response to one of those red dots in there. But then the virus copies itself, makes a huge mess of itself, and a bunch of those mutations are covering up the red spot. And so those are the viruses that are going to then proliferate in the face of that immune response that's trying to neutralize the red spot. And so you get viral escape very easily from most HIV vaccines. And so this is the key scientific problem we have for HIV vaccines. Next slide, please. So what do we, here's the pipeline for efficacy trials. There's only been a small number of large HIV vaccine efficacy trials, about 10 or less than 10, you can compare it to COVID and see, how many large trials we're having for COVID. So that's really a shame. In the past, on the left side of the graph, prior to 2013, well, really almost all of these trials, the approach has been empirical. Make a vaccine that makes, and see what kind of immune response it makes, and hope that it might be effective in neutralizing, blocking the virus in some way. So empirical, you keep trying, you keep revising. So we had the AIDS Vax trials that was made of a GP120, just the spike protein. So that one didn't work. So then RV144 was the Thai trial that you may remember, that used also a protein, but also a non-pathogenic viral vector, an ALVAC vaccine with a protein boost. And there was some hint that maybe it works and low level of efficacy. And so for years, the vaccine field built on that idea of let's take this ALVAC non-pathogenic virus that delivers an HIV antigen, the protein boost, let's make it better. So that's the Pambili and the HV10505, sorry, the Pambili trial, excuse me, not the HV10505, was trying to make the RV144, redesign it and try and get a better antibody response. Merck, the pharma company in 2005, had a very nice vaccine concept with a non-replicating adenoviral 5 vaccine. And that was when there was, at that time, a lot of pharma involvement in HIV vaccine development, or a fair amount. And that trial also did not show any efficacy. And there was a hint that maybe it caused an increase in the risk of HIV with the Ad5 vector. So there have been a lot of stumbling blocks along the way. None of these vaccines ever were able to induce neutralizing antibody that was broad, meaning that it could neutralize many different types of viruses. The neutralizing antibody response that was developed, that evolved from these vaccines, was very narrow and it did not prevent HIV. So where are we now in this? You can see in this graph here on the right-hand side of the timeline, 2017 and onward, there's been a bunch of new trials where, with a renewed interest from the US government funding, there's been renewed interest from pharma companies with J&J, Janssen coming forward, but still the concepts that they were testing were somewhat empirical and older concepts. So you can see, just ignore the top two in the green box because those are actually passive antibody trials that we'll talk about. Those are not vaccines. So HV10702 and HV10705, I'm going to talk about those two studies, which recently this past year, actually within the past month, came out saying they were not effective. Let's talk about those a little bit. The next slide, please. So HV10702, this trial in 2020 released data showing there was no benefit to it. This was a very large trial. Many of these trials are conducted in Southern African countries now, where the rate incidence of HIV is still quite high. And this was, again, that ALVAC, non-pathogenic virus used to introduce an HIV antigen with a protein boost of the HIV envelope spike. So still it's that old concept that was known not to induce broad neutralizing antibodies, but the field thought, well, if we can just make it better and make it a subtype C so it matches the subtype of infections in the study population, maybe it can work. And we knew it was a non-neutralizing antibody type vaccine. And the results basically show at the Kaplan-Meier on the right that the vaccine and placebo groups had the same rate of infection. So there was no benefit. So that was a big blow because this was taking that RV144 Thai trial concept and trying to make it better and focused in a South Africa population. Next slide, please. So the other big disappointment, which just came last month, was J&J had made a huge investment in another technology for an HIV vaccine. And this one was also with a non-pathogenic virus, an Ad26. It's actually the same platform that J&J uses on its COVID vaccine, which did show efficacy, and their Ebola vaccine, which also showed efficacy. And so here they've inserted HIV antigens, what are called mosaic antigens. They took antigens across many different viral subtypes, clades, and different viruses, thinking that would give you a broader immune response. Still, they knew it did not give neutralizing antibodies. They saw that in earlier studies and in animal. They knew it wasn't going to give neutralizing antibodies. So it's still testing that older concept. But still, the thinking was, if we can make it better, make it clade C specific, and it was tested in two studies. One, the Imbokodo study, was in women in Southern African countries, women at risk of HIV. The other trial, still ongoing, called Mosaico, that's in MSM and transgender women in the Americas, and that study is ongoing. The vaccine is a little bit different, so they're still continuing that study. But I'm just showing the results of the Imbokodo study here that were just released, and they've showed 25% efficacy, which could have basically been zero. So that's really not statistically meaningful. So I think what this is telling us, I think it's pretty clear that we need to have the non-neutralizing antibodies aren't going to work. We need neutralizing antibodies. So next slide, please. So I think that many people in the field, most groups in the field have been working on neutralizing antibodies for decades. And so even though these trials were going forward, people were still working on how can we induce broadly neutralizing antibodies. And this was the study that IAVI did when I was there, just screening people with HIV all around the world and many different continents to see, are there people that make antibodies? People make antibodies to HIV, even though it doesn't protect them or doesn't help treat them. But people test positive on HIV test, you're making antibodies. Are those neutralizing and broad? Could we use some of those antibodies to help us figure out how to make a better vaccine? So we found after testing a whole number of people that about 1% of people with HIV do produce neutralizing antibodies that are very broad and potent across different clades. We call these elite neutralizers. So next slide, please. And so this is showing that what happens to people who are infected with HIV, about 20% of chronically infected people actually get broad neutralizing antibodies over time. So initially you have a binding antibody to HIV, and then over time your immune system starts to develop to mature that antibody response and make it more specific for the viral strains that are circulating in your infection. And then in some people, they actually develop antibodies that can neutralize across the different strains that the person is infected with. Now, it doesn't actually help them, their infection. But you can isolate those antibodies from blood and you can test them across other viruses in the lab. Next slide, please. So this research has been very important for us to understand how do broadly neutralizing antibodies develop in natural infection, and what are the stages it takes and how many years, how does that happen? How can we replicate that with a vaccine? How can we give one or more vaccines that give an antibody response and then another vaccine to push it along the antibody response along the pathway till it becomes broad and effective? Next slide, please. So the idea is to take what we're learning about that and then try and design vaccines to then to give the right response that we're looking for, those steps. And that's basically called, rational vaccine design. Instead of just empirically trying things, we know what we want. We want to get this broad antibody. VRC01 is one that people may have heard of that was identified by the NIH, an antibody that's very broad. It was identified from an HIV-infected person. It could neutralize across a number of viruses, very, very, very resistant viruses. And then that was characterized, that antibody was very carefully characterized. So that's the goal. That's what we want to induce. Now, how can we make a vaccine that can get us to that antibody? So it's much more rational, the design. So I won't talk about all these concepts, but these are different concepts in rational vaccine design to get the body to produce broadly neutralizing antibodies, but they're all very early stage. They're basically in the lab or in phase 1 studies. So you can see there's a big gap from these large efficacy trials that just finished and failed, and this next generation that we're going to have to wait a while to figure out how to make these broadly neutralizing antibody vaccines. So next slide, please. So in the meantime, because these neutralizing antibodies have been identified, a number of different ones besides VRC01, the idea is, well, why don't we give these as passive immunization? So you usually give an IV, but they actually can be formulated to be injectable, like cabotegravir. Could you give these, and could you engineer the antibodies so that they're safe and could last like 3 months? The level in the bloodstream could be good enough for 3 months to protect you. So that concept is now being tested in a number of studies. It's not a vaccine. It's a short-term protection that maybe could get groups that are at high risk through a period of years until we get a vaccine. And the concept is being pushed so much that the idea of, can we get it so the injections can be less than 3 months? Can you do an injection every 6 months of maybe 3 really potent combination antibodies? that then would protect someone for 6 months? And could it be like an injection, like people might get Depo-Provera for contraception? Could it be something that actually could be done in a widespread way? So that's what people are working on now. And this is the concept of passive immunization. So I just want to show you a couple of trials that you probably heard about, the AMP trials. Next slide, please. So these were trying to test this concept. Can you give an antibody, IV in this case, and prevent HIV? So this was a large series of studies done by the NIH in the US and in the Americas and Africa, in the one trial in women and one trial in MSM and transgender women. So the two trials basically gave IV, one IV monoclonal antibody, this VRC01, every 2 months. So people had to come into the clinic every 2 months and get an IV infusion. Very, very, very difficult trial, very complicated. And then they followed to see what was the rate of HIV infection. And people were given PrEP as well, and all the other standard of care. And basically, the results were pretty much negative. That, well, if you were exposed to a VRC01 sensitive virus, then this infusion could protect you, but it did not protect broadly against other viruses. So next slide, please. This is just showing. So sorry, there's a lot of numbers on here, but basically what it's showing is what is the prevention efficacy depending on the sensitivity of the virus that the volunteer in the study might have been exposed to, through their own behavior. So basically what it's showing is that if you had a very resistant virus, and that's the bottom line, that dark blue, so it took a lot of the antibody, over 3 micrograms per mil of the antibody concentration to neutralize the virus. If you had a difficult virus that you were exposed to or infected with, there was no efficacy. It was basically around 0. If you had a virus that VRC was really good at neutralizing, that's on the top, there was some benefit. So there's a 75% protection for those people, but that's not going to be a product then that you can use broadly, because you have no way of knowing if you're in this risky situation of being exposed, the sensitivity of the virus, that's not something that people know. But what it showed is that the concept could work. You just need better antibody. And so now the idea is to take 3 antibodies that have more breadth overall, and you can give smaller amounts. So that's now proceeding in clinical studies. Next slide, please. Like I was saying, it was disappointing that these studies didn't show protection off the bat, but VRC was just the first antibody that was available to test. It was really a proof of concept, and it gave a signal that broadly neutralizing antibodies can work. And also it helped the vaccine field realize, we're going to need to induce broadly neutralizing antibodies. So the vaccine trials have shown us that. These studies have shown us that. And it also helped give an idea of what concentration in the blood is needed to block HIV when you have an antibody. So those are all really helpful things for the vaccine field. And the next slide is just showing, also should be very hopeful, because there's a lot of other broadly neutralizing antibodies out there that have been well characterized and are in trials now in combination. And some are in phase 2. So again, it's not a vaccine, but this is a technology that actually, and there's been a lot of work done on costing and access. The Bill and Melinda Gates Foundation is a big funder of this activity, because they want to see it as an accessible, low-cost technology that could be deployed widely. That's the goal for the next few years, or till we can get a vaccine. Next slide, please. So this is the last one I have. We didn't really talk about this today. I don't think I didn't come to all the talks. I missed a few, so I'm sorry if this is a repetition at all. I just wanted to mention what's known about COVID vaccination and people living with HIV. So HIV has been shown now, and we have more data, that's an independent risk factor for increased COVID hospitalization and mortality. It's not as huge a risk factor as some of the other ones, particularly if it's treated and viral load suppressed HIV, but it is an independent risk factor, even on ART. So there is a real reason to make sure we have the right COVID vaccine regimens for people living with HIV, either on ART or even as well as uncontrolled HIV. But there's really not a lot of data that people living with HIV were included in many of the trials, the efficacy trials, which is good, but the numbers were very small. The one study that had quite a lot of people with HIV in it was the Novavax efficacy trial done in South Africa. They actually didn't test people for HIV before they went in. They tested them later. Well, that wasn't a criteria for going in to have an HIV status, but so the data they found was that the vaccine was 50% effective among all adults, but in the people who later just tested negative, it was 60% effective. So that suggests that people with HIV, and most of these people were on ART, have the vaccine may be less efficacious. So that needs to be looked at. And there are groups like CEPI that are trying to fund some follow-up studies now to figure out what are the best regimens. Should people with HIV maybe need an extra boost? Are certain vaccines better for them than others? But right now, the recommendation is that everyone with HIV should be vaccinated. There's no specific safety concerns specific to people with HIV for COVID vaccines. There are general safety issues with every vaccine, of course. In the US now, they're recommending a booster for immunosuppressed populations, and they are including people with HIV with CD4 counts less than 200. But in practice, I've heard that basically people living with HIV are eligible for a booster in the US and perhaps the UK. But again, that's based really on not much data. So I just want to give that little summary. I mean, the answer is, COVID vaccination for people living with HIV, the answer is yes. So even though I've put all these kind of caveats, the answer is yes, yes, and yes. So, okay, I think that's all I have. I just wanted to, mention a few lessons learned from COVID vaccines. I think HIV is really a formidable target. We knew it was, but then seeing the success of COVID vaccines, you can see that, the same people, actually the same labs, a lot of the same groups that made the COVID vaccines have been working on HIV for years. So it's a very, very challenging target. It's not just because there hasn't been enough money. We've seen that vaccine development can be fast if we can figure out these challenges. And the pharmaceutical industry is really important. I mean, we need to have them as partners for HIV vaccines. Safety is always important. And the long-term investment in basic research, which may seem kind of dry and abstract, we've seen in HIV field how important it's been for development of treatment. And that's the same for vaccines. And then the global view is, we know that in the HIV field, the global view is really important. I think we've known that, and you can see that again from COVID vaccines. No person is an island anymore. So not even New Zealand. So, okay, next slide, please. Thanks very much for the time. I thank these people shown here for sharing their slides with me, and I really appreciate the invitation, and thanks also for the support of the funders for this day. And I just want to give a shout out to IAVI, which I don't work there anymore, but they're still fighting the good fight for a vaccine. Thank you.
That was great. Thank you, Fran. That was really informative, and a really broad insight into the history of vaccines and the possibilities. I think the BNABs, body neutralizing antibodies, I think are really interesting, and there's lots of potential there. So it's good that they're, it's a glass half full kind of perspective in terms of those and what might be able to come from those. One of the questions from the chat is, do you think that mRNA-based HIV vaccines will be able to bypass the HIV diversity with the success they've had in the COVID space?
Yeah, I think it's not going to be as simple as it may seem from COVID, because an mRNA vaccine can give a great immune response, perhaps, but getting the breadth. So far, the only understanding about how to get breadth is to do the sequential immunization. So one of the mRNA companies, Moderna, is actually working on an HIV vaccine using that rational approach with IAVI. And they have the first 2 different vaccines they're going to put in a phase 1 trial, where the first step to get the first stage towards neutralizing antibody, then a second vaccine, which is different, still mRNA, but a different insert to get to then push the antibodies the second step. But I was working on that project at IAVI before I came here, but the scientist who developed the concept thinks there's at least 4 steps, at least. So you'd have to make 4 different vaccines. So the concept is not ready yet. It needs still further work and development. And so I think it could work. mRNA could help it along because it's faster and cheaper to make the different vaccines with mRNA. So that could be really helpful.
That's great. And just the challenges you presented by doing infusions, I think is that'd be quite the challenge in terms of doing studies and research, but also in terms of ongoing utilization. So I think the cabotegravir long-acting injectable kind of model with those, or the injectable things that they put under your skin that slow release, might be potentially options for those in terms of,
and I think people are also thinking about the implants for BNAbs. Could you put, so they're putting I think islatravir, the Merck long acting, they have a pill, like a monthly pill, that's I think in a phase 1, and then they're looking to put it into their contraceptive. They have a contraceptive implant, Organon, so that would be mainly for women, but then people have to accept having an implant, so that's not highly acceptable broadly. But it could be that I think that some populations that are impacted by HIV, in some cases, some of them tend to be early adopter populations. We used to work with this population of MSM in Eastern Africa, and we did a PrEP study there, and they actually had a lot of interest in some of the more novel methods for adherence, and they were very interested in like using apps and using different little pill things to measure your pill adherence. And so I do think we shouldn't rule out complicated or early adoption kind of stuff, because some communities may actually be interested in that.
It's very much a motivation thing. So what are people motivated to do in terms of that protection piece, and how far will they go? And a lot of people are very motivated. So so there's no more questions from the chat, but I'd really like to thank you for that. That was very insightful and very easy to understand. I think you did a really good job of presenting a very difficult topic for a lot of people. So thank you for that. So
thanks so much. Take care.
That was great. Thank you.
Thank you.